Imagine that a scientist studying a genetic disorder like cystic fibrosis identifies a potential mutation in the CFTR gene from patient's DNA sample. Before they may conclude that this mutation is responsible for the disease, the scientist needs to validate it by comparing it with known mutations and checking if it disrupts gene function and determining its frequency in affected versus healthy populations. Validation involves confirming the mutation's presence, determining its effect on gene expression or protein activity, and linking these changes to observed phenotypic differences. There are various strategies for validating gene mutations, and they fall into several categories. There are experiments that can be performed outside of living systems, whole systems, that are called in vitro studies. Then there are those that are done in animal systems. These are called in vivo studies. There are also functional genomics that can be performed. Then there are biochemical and molecular assays. We also have computational approaches that can be used to study the impact of a gene mutation, as well as omix approaches. So this is where we are looking at things like proteomics, but it can also include metabolomics and transcriptomics. So let's look at each of these in detail. The first one in vitro studies, or studies that are performed outside of whole living organisms, is cell culture models. Here, mutations can be introduced into cell lines using techniques like CRISPR-Cas9 or plasmid transfection. Here, the cells can then be studied for changes in phenotype, so that's how they look, their morphology. Then they can also be assessed for gene expression or protein function. And here you can use qPCR to look at genes, as well as looking at whether the cell line becomes drug resistant, for example. Under the in vitro studies, there are also reporter assays. So here you use reporter genes like green fluorescent protein or luciferase that are linked to the mutated gene's promoter or regulatory elements, and these can then be used to assess the impact on gene expression. A third experiment that can be done under the in vitro studies is protein expression and purification. Here you express and purify the mutant protein or proteins in bacteria or mammalian systems, and this allows for in-depth biochemical and biophysical analysis, such as binding affinity studies or enzyme kinetics. But first, you express the protein and purify it. Now we can look at a second method for studying gene mutations, and this one is in vivo studies or inside whole living system studies. So typically you have animal models. Here, genetically engineered animals such as mice, zebrafish, or Drosophila can be created to carry the mutation. Once these animals carry the mutation, you can observe any phenotypic consequences, so how they look structurally, morphologically. If there are any differences, you will be able to observe it once you express these genes in the animals. Observing these phenotypic consequences in the models provides insights into the mutation's impact on organismal development, physiology, and behavior. Now we can have a look at functional genomics approaches. So in this approach, you may use CRISPR screens, for example. And here, high throughput CRISPR screens can be used to identify genes that, when knocked out, modify the phenotype caused by the mutation, assuming you've identified a phenotype caused by the mutation. This can help place the mutation in a broader biological pathway or network. Under the functional genomics approaches, you can use RNA interference knockdown. So here, RNA interference can be used to reduce the expression of the gene that's carrying the mutation in order to assess its impact on cellular functions. There are also biochemical and molecular assays. The types of biochemical and molecular assays that are available include enzyme activity assays, and you have to determine if this is suitable in your context. But in enzyme activity assays, if the mutated gene encodes an enzyme, then assays can be used to measure changes in its catalytic activity. There are also binding studies, and here techniques like co-immunoprecipitation, surface plasmon resonance, or yeast 2 hybrid assays can be employed to study changes in protein, or protein DNA, RNA interactions. Under the biochemical and molecular assays, there are structural studies that can be performed, and here X-ray crystallography, NMR spectroscopy, or cryo-electron microscopy can be used to determine if the mutation causes structural changes in the protein. There are computational approaches as a fifth way to validate gene mutations. In computational approaches, you may perform molecular dynamics simulations, and these can predict how a mutation might alter protein structure and dynamics. There are also in silico functional prediction tools like SIFT, Polyphen, or CAD can predict the potential impact of mutations on protein function based on evolutionary conservation and other criteria. The sixth approach that may be used to validate a mutated gene includes the omix approaches. And here, with transcriptomics falling under this category, you may use RNA sequencing, which can reveal how the mutation affects gene expression profiles. Or you may use proteomics. And here, mass spectrometry-based proteomics can be used to analyze changes in protein expression, post-translational modifications, or interaction networks. Metabolomics also falls under the omics approaches, and here you analyze changes in metabolite profiles, and these can provide insight into the metabolic consequences of the mutation. So each of these strategies provides different types of information, and often multiple approaches are combined to comprehensively validate and understand the functional impact of mutations identified through genomic sequencing. Thank you for checking out this video. Thank you to all the new subscribers and all my existing subscribers, and I will see you in the next video. Bye.